Hi, everyone. Welcome, welcome. My name is Dominique Cruz, and I'm the project manager for the Rare Disease Cures Accelerator Data and Analytics Platform, or as we like to call it, RDCA-DAP. RDCA-DAP is a program of Critical Path Institute and includes a platform to aggregate rare disease data and facilitate data sharing and analysis to users worldwide. And we also operate as a program working to leverage rare disease data to facilitate the development of new drugs for patients. And we like to host this webinar series. This is actually our last one of 2023, and we're super excited to welcome everyone to today's webinar, VCP Diseases, an Integrated Approach to Neurodegenerative Disorders. I'm going to start with just a few housekeeping items. If you have any questions throughout the presentation for our speakers or the panelists at the end, or about the program in general, please put those in the Q&A chat box. The chat function is disabled for everyone, so we can make sure that we aren't missing any questions that pop up. So if you have any questions, please put them in the Q&A box. Also, just to note, all the participant lines are muted so we can reduce background noise. We're also recording this presentation, so it will be available for on-demand viewing via our YouTube channel shortly after the presentation to everyone who registered. We've also included a general contact email address. If you have any questions or want further information after the webinar or to connect with any of our speakers or panelists, please feel free to contact us there. At this point, I'm pleased to introduce our presenter. Dr. Amel Pindon is the Chief Science Officer at Cure VCP Disease, Inc. Throughout her career, she has focused on understanding mechanisms in neurodegeneration and neuronal lesions and translating this knowledge into discovering treatments. And at this point, I'm happy to turn it over to Dr. Pindon for her presentation.
Thank you. So I'm going to share my screen. So I would like to, hello everyone, I would like to thank the Critical Path Institute for giving me the opportunity to talk about VCP disease and the challenge encountered by heterogeneous rare diseases. So in this webinar is going to address different aspects of degeneration, starting from presenting you the Prince of Proteus vasis, to think about of heterogeneity in an unconventional manner, and asking the questions to be ultra rare or not to be ultra. We'll open the debate on how to get a mechanistic integrated approach to degeneration. So VCP stands for valosine containing protein. It's an AAA class of ATPase containing 3 domains, the N-terminal, where most cofactors and substrates bind, and the D1 and D2 domain have the ATPase activity, allowing changing in the conformation and movement that will process the substrate through the homohexameric structure. VCP has over 40 cofactors that will confer multiple cellular function to VCP. It has different names such as segregase, chaperone, or unfoldase, and depending on the subcellular localization of VCP, it will trigger different functions. So the purpose of this slide is not for you to read every detail, but just have a general view of the numerous cofactors and function. For example, when VCP is located in the nucleus, it forms a complex with, for example, UFD1 and PL4, and will trigger double-strand DNA repair, cell cycle regulation, and spoiler alert, some very recent discovery will be reported by several groups at our scientific conference in February, showing the importance of VCP role in the nuclei. In the cytoplasm, it will form complexes with pink one, WASH-C4, several types of UBX, P47, etc., that will involve a process such as ERAD, ELDR, autophagy, mitochondrial and lysosomal dysfunction, and stress granule disassembly, just for example. And when VCP is localized at the membrane, it will promote dendritic arborization by recruiting neurofibromin and then bind to ATL1. The complex of cofactor binding with VCP are present in most of the form of neurodegenerations linked to autophagy processes. Becklin-1, PINK1, Huntington, optineurin, tau, TDP43, SOD1. So focusing on autophagy and neurodegeneration, you can see really the involvement of VCP. If you look at the key features of neurodegeneration, we can see that outside of inflammation, they are all common with VCP function. You have impaired autophagy, impaired protein clearance, impaired complex disassembly, forced cell cycle reentry, and post-mitotic neurons, mitochondrial and lysosomal dysfunction. If you look at VCP key components of the different process where its key components, it's degradation of aggregate-prone proteins, granulophagy, for example, stress granule, complex disassembly in cytoplasm, but also in the nucleus, TDP43 localization and aggregation, what was the first aggregates shown, autophagy and lysosomal function, mitochondrial fusion and fission, cell cycle regulation, double-strand DNA repair, and dendritic arborization. So you can see that really VCP is really at the center of so many different components of degeneration. And VCP, what is interesting, it has been shown to seed the aggregates, so move from one cell to another. It has been shown in ataxin-3, for Huntington, and for TDP43. So you can imagine how the disruption of VCP function leads to neural, but also muscular degeneration. There are two ways to have disruption: by mutation of VCP, the first one, which historically was identified as VCP disease, renamed multisystem proteinopathy 1, what also called as IBM-PFD, because it regroups the patients that have the three manifestations: inclusion body myopathy associated with Paget disease of bones and frontotemporal dementia. They are all highly TDP43 pathology associated. This has a very underestimated prevalence of 1/300,000 people, but it has now been found that VCP mutation trigger way more than IBM-PFD, and it represents 1 to 2% of familial ALS, 0.2 to 0.8% of sporadic ALS, which is already there a few thousand patients. Also, CMT2Y are exclusively VCP patients, and we find patients with Parkinson's disease, with HSP, vascular tauopathy are also specific to VCP, Lewy body dementia, and it has recently published that there is a pediatric cohort of patients with neurodevelopmental disorders. So the manifestations of VCP can be cumulative or single manifestations, which add to the complexity and the heterogeneity of the disease. The other way of disrupting VCP function is through the mutant impeding the function of VCP. So through the polyQ type of mutation that traps VCP, as for Huntington, ataxin-3, and other types, and they were shown to have, for example, a significant decrease in DNA repair. The same observation was done for poly-GA with a study on C9orf. Another type of VCP function modification, for example, is for ATP7A mutation that can trigger the ability for the protein to bind to VCP when that property is not present with the wild type protein. Oops, went too far. So the arguments in the scientific community is whether VCP needs to be activated or inhibited to treat degeneration. It might not be as simple as a yes or no answer. Some cases clearly show the benefit of increasing VCP function, but there are findings also that are contradictory. I think we should pay attention to the pharmacochemistry of the inhibitors used in these studies, depending on whether the compounds are allosteric inhibitors, partial inhibitors, or competitors specific to the D1 domain or the D2 domain, or compounds that are don't alter the ATPase activity per se, but prevent cofactor binding. The complexity and difference in pharmacological compound might lead to misinterpretation of biological data. Our reference type of manifestation, VCP disease or MSP1, is shown to activating VCP rescues the wild type localization of TDP43 and triggers TDP43 aggregates clearance. Same as for mutant SOD1 in ALS, where an overexpression of VCP restores the balance between the soluble and the insoluble fraction of SOD1, and an inhibition of VCP increases the imbalance seen in ALS. Or, for example, VCP activation might be the way to rescue tauopathies, where VCP disaggregates amyloid-like tau fibrils. On the other hand, in ataxia type 3, targeting VCP binding motif of ataxin-3 improve phenotypes in Drosophila models of spinocerebellar ataxia type 3. In Huntington, VCP is trapped at the mitochondrial membrane, and suppressing VCP mutant Huntingtin binding reduce Huntington disease-related behavioral and pathological phenotypes in 2 different transgenic mouse models. And you can see restores the neuron survival rate. The question might not be to activate or inhibit, but to increase the VCP function in the distribution of key proteins between the nucleus and the cytoplasm, such as TFEB or TDP43. Activate VCP capacity could be seen to increase the protein clearance and autophagy. And in some cases, we could combine it with the inhibiting VCP interaction with the mutated protein, which prolonge their binding with VCP, forms aggregates, sequester VCP, and triggers nuclear redistribution, as it happens with TDP43, but not really per se inhibits the ATPase function of VCP. So what do we do about it? Cure VCP disease was created only 5 years ago by a few patients getting together, including our CEO, Nathan Peck, and his wife, Alison Peck. It is dedicated to all patients with a VCP mutation. We are very young and very small, but very dynamic and driven. We have a medical advisory board and a drug discovery scientific advisory board to fulfill our mission and vision focus. And everything started with the creation of the patient registry covering several countries and growing. Our strategy is based on 4 pillars: the preclinical toolbox, gather assays, patient-derived models, and in vivo model. The clinical readiness includes the creation of new outcome measures in the real world and more relevant to the patient. the longitudinal natural history studies, and biomarker studies. The third pillar is the treatment discovery and includes screening small molecules and 2 different techniques and projects on gene editing. The regulatory and industry pillar, the last one, includes gathering data from patients through the registry, several surveys, panels, and posters presented by the patients to the scientific community. And we also have more traditional engagements program for the scientific and the patient community, such as conferences. So Nathan likes to say we have moved fast in 5 years. I will just attract your attention on a few milestones: the registry, the FDA listening session, the retrospective and prospective natural history study, and the VCP standard of care publication orchestrated by Cure VCP disease. I have to say that all that was performed. We could do that because we have the CZI Rare as One project. We are grantees from the second part. So in 2023, we developed our 4 pillars with the creation of iPSCs models, patient-derived from 2 different patients with different manifestation characteristics. We have the R155H, which is the most common mutation among the patients, and the R159C. The iPSCs were made from PBNC, and we have reprogrammed them in motor neurons and myoblasts that are able to differentiate in myotubes. So we have assembled a task force of different labs that are involved in the characterization and assay validation on those iPSC models. We have started our prime gene editing project in partnership with the Genethon. In that project, we have created different type of cellular models according to the stage of the project. We are screening pegRNA, and we will on HEC293 engineer cells, for example, but we will validate at the end on our iPSCs and myoblast patient-derived models. We have also concluded the longitudinal natural history study that was performed at Nationwide Children's Hospital. I will talk more in details about that. And we have also started a biomarker on patients' blood that were collected. The samples were collected during the longitudinal natural history study. For 2024, we just started a collaboration with NCATS to develop 3D organoid models. Muscle 3D printing, so the technology is in place, and we are going to use our patient-derived myoblasts. And the new technology of neuromuscular junction is going to be developed also with our patients' cells. And this will be used for screening on the organoids to start with 2,500 compounds. So to summarize a bit what we do, we have this preclinical toolbox where we gather the assays, cellular models, and we have animal models that are in academic labs on Drosophila and different mouse models. And we are starting to gather reference compound to have a chemical library to facilitate screening. The longitudinal natural history study was conducted after, so that's the clinical readiness, the second pillar, sorry. So the longitudinal natural history study was conducted after 2 years because the functional measures showed significant changes already over 1 year. And the rate of progression was sufficient to measure endpoints. We have developed new outcome measures for more relevant to the patients based on everyday life tasks, such as get up from the couch, pick up a sock from the floor, put on a shirt, etc. The scorecards were created and the baseline assessments are completed. So I told you that we have a biomarker study that is being performed on blood from patients who participated in the natural history study. And we are analyzing at time 0, 12, and 24 months. The urine samples were also collected and will be analyzed when our finance will allow. All these data should help us create a master protocol for clinical trial. So in summary, the application for VCP, which is also called P97, more in the oncology part, it's more called P97. So we have a direct interest in 6 neurological and mitochondrial associated disease I mentioned before. We have evidence base for CSA3, Huntington disease, ALS, and vascular tauopathy. There's also a general implication in degeneration and aging, specifically neuron and muscular degeneration. We have non-neuromuscular disorder application in testing, especially in oncology, in ophthalmology, and in virology. VCP is also a biomarker for psoriatic patients, and there's a recent publication for 2 days ago that shows that VCP is recruiting neutrophil in psoriasis. VCP is also a biomarker of different cancers, and specifically sarcoma. So one of our issues, as many other rare diseases, is the heterogeneity of our patients' manifestation and misdiagnosis. We are proposing to address heterogeneous rare disease by shifting from symptomatic to mechanistic approach across rare diseases. When patients are in silos according to symptoms, we miss opportunities for treatment and patient identification. So the integrated approach will not address the effect, but the cause. The problem is how to identify the common cause. For us, it will be to identify the VCP patient that have a mutation, and they might be hidden in the different diseases or with the wrong diagnosis. As you remember, I mentioned that it's in VCP patients are found in so many different classification of diseases. The diagnosis confirmation is based on genetic testing. So we are using now predefined panels with neuromuscular disorder, frontotemporal dementia, and ALS. But if we want to identify more patients, should we expand those panels to all the movement disorders, mitochondrial disorders, and all types of dementia or even lysosomal disorders? So that might not be the ideal approach for us. So another approach is through metadata analysis, as in CPAS and RDCA-DAP has developed. So it can be used for different possibilities, finding commonality in unconventional symptoms, identifying new relevant and tailored endpoints instead of sticking to more general muscular disorders. Maybe we can really identify the right outcome measures for VCP patients. We are thinking of common biomarker across rare diseases. And one of the major issue for a lot of different rare diseases is to improve the variant classification. The VUS is also a big issue. And I think this data analysis through CPAS and RDCA-DAP might help to better define variant classification. We can also think about identifying gene modifiers with the ontology that can be created through this platform. And we know now that VCP is a good candidate for as a gene modifier for NF1. We have data on Huntington and VCP, Ataxin2 and VCP, TREM2, and one or two patients with ApoE4 and VCP. So the other very interesting data collection application would be the modeling disease progression, but with such an heterogeneity for our disease, but also for many other rare diseases, is how to model disease progression in those cases. Can we get to try simulation across diseases, trying to pick, in fact, in the different diseases, a model disease progression. So to gather all this data, we stimulate the scientific community to share their data. We have monthly virtual scientific focus group where people are really sharing their data, and there is very nice and dynamic discussion and brainstorming every month. And we will have our international scientific conference in February in Caltech, where we are going to try to think about this cross-disease integrated approach for VCP applications. And we are going to look at VCP from oncology up to neuromuscular disorders. We will have presentations, roundtables that are not limited to VCP, but really addressing issues that are common to all rare diseases and late onset diseases. And we'll have posters, as I mentioned before, we will have patients presenting posters to the scientists with their history, the history of their heredity in their family, and the different symptoms, maybe symptoms that are not conventional for neuromuscular disorders, and we hope that it will trigger the scientists to will pick their brain and maybe have new theories based on that. So these are all the references. I'm sorry that I didn't put in every data that I shown. So that's I ran through the presentation. I talked very fast. I'm sorry. That's it for the VCP, and I think now I'm going to leave. Let's.
Yes, well, thank you, Armelle, for the great presentation. And just to remind our audience, we're heading into a Q&A now, so we'll start with a few discussion. I'm welcoming here Terina Martinez from our team. I think it's the first time that I ever introduced Terina publicly, so welcome, Terina. Terina is the Executive Director for our Consortium for Rare Ataxias, and also Executive Director for the HD Risk, or Consortium for Huntington disease. Terina is a specialist in neurodegenerative and rare neurodegenerative disease. So great to have you here, Terina. And maybe as a start, I'm sure like the presentation triggered a lot of questions and reactions. So if you'd like to comment, make an opening statement or comment on some of your reactions to what Armelle just presented, and maybe if you have some burning questions for Armelle, you can kickstart our Q&A.
Sure. Thanks, Alex, for the introduction. And it's my pleasure to participate in this webinar today. I've learned so much listening to Armelle walk us through the complexities of VCP disease. And of course, as you mentioned, Alex, with my role in our spinocerebellar ataxia consortium and also Huntington's disease, my mind is just blown on the interconnectivity and the fact that we know that the diagnosis odyssey for many patients goes a long time without being properly diagnosed. And I think that, a lot of the points that Armel mentioned about, potentially expanding the genetic testing, really more understanding better the pathways of the disease and the really complex convergent cause with divergent effects, and the integrated approach that Armel illustrated, I think is, first of all, an excellent approach for VCP disease, but also understanding its connectivity to these other pathologies. So I just wanted to emphasize that. And one question that I have is actually, kind of coming back to, actually, it's my question for Armel, and then maybe I can follow up on it too. Because of the extreme complexity and heterogeneity in VCP diseases, how much of that do you think, or is this going to be one of those, all of the above potential combination answers? How much of the complexity of these convergent cause, divergent effects is because the measurements aren't precise enough, or the understanding of the biology and pathology isn't granular enough? Or do you think it's actually due to the heterogeneity on an individual patient basis is actually biologically driven because of the upstream cause-effect paradox? I just want to see what your thinking is there.
I think that the gene modifier is really a question here. The whole genome sequencing and really comparing genome in between the patients might be a way. The issue is that we have very limited population, so to be significant when we compare whole genome sequencing will be more of a challenge. But it's true that for the main mutation, that the most common mutation that we have, the 155H, we have patients with muscle issues and other patients with just frontotemporal dementia. So really, I think that the gene modifiers is something. The epigenetic is also very important to characterize that. And I think the organoids are going also to give us some answers, comparing muscles, really real muscles, 3D muscles, from one patient to another with the same mutation. And we have also with the same mutation patient that have only muscles degeneration, and other that have motor neurons affected, and other that have just the frontotemporal dementia. So it's we are at the beginning. We understand more and more that VCP is in fact triggers aggregation. So it can trigger different type of aggregation, like the tau, the TDP43. So discovering what type of aggregates are going to be triggered will be a key aspect of the future for VCP, because why some of them have the aggregation more in the brain than in the muscles. Why TDP43 in some cases, why tau in other cases. Well, so that's going to be a real questions. The general thing is, in fact, the redistribution of key proteins between the nucleus and the cytoplasm, which is general to all type of VCP mutation. So maybe addressing the disease through this redistribution, whatever the type of aggregate, will be something, a way to go. I don't know if I answer properly your questions.
Yes, that was wonderful. Because like you said, I think it illustrates the importance of the genetic modification, epigenetics. I think that's also really key. And of course, anytime maybe I should let Alex ask the question, but you've got what you said got me really interested in another topic about, again, the complexity and the interplay between gene environment, epigenetics, gene modifiers. Have you explored, or is this something that you're looking at in future efforts, the complexity of race, ethnicity, geography, and how that can be, having a role here?
So we have more patients that have only FTD in the Hispanic population. But that's the only difference in ethnicity that we have seen up to now. And for the diversity, for the ALS, or we didn't see any ethnicity linked modification. And your question was, sorry, the sound is really bad. Your question was about diversity in the natural history study. So just for the ethnicity, or you mean the diversity in the phenotypes?
Yeah, it's interesting because I think it could go either way. I mean, you've already addressed part of it by describing the enrichment of, or the greater frequency or incidence of the just FTD representation among the Hispanic population. But I was curious if you, so it seems like you probably derived this from the natural history study. But are there other directions that you think are emerging from what you're seeing in that natural history study that would further elucidate potential contributions from race, ethnicity? Because I know in the rare disease space, it's always difficult to capture diversity effects because you're already looking in a very rare, narrow population. So I just wondered if you had other directions that you're ready to explore, or if you need more opportunity to coordinate these screening efforts and other approaches that you described.
Yeah, so for diversity, I think it's an issue, as you said, for all rare diseases, because we are already we have very few patients. And I think there is a discrepancy between the patients that are going to be tested in different population, because there is the more educated population you have, the more genetic testing you have. So that's already a bias from the beginning, because underrepresented population have a tendency of not trusting the system and not willing to be diagnosed or tested. So we have very few patients from underrepresented populations. African American, we know that there are some patients that are there, but they don't want to be tested. Maybe because there is also this view of this disease as a curse, as says Nathan, and there is no cure. So what's the point to be diagnosed? So that creates also a discrepancy between the different ethnicity because of the level of education. So that's maybe in rare disease, that's a common issue to many different people. And there is also some maybe blockage from the part of clinicians who don't want to do genetic testing, because what's the point? And that's where we really need to do to educate clinicians and physicians in general to use more genetic testing. In our case, the question of where should we be on the panels also, should we expand the panels, because the disease is so heterogeneous. We might have some patient in lysosomal disorder or mitochondrial disorders that we don't know of, like this new pediatric population that we didn't know about only 3 years ago. So that's I have hope that the genetic testing will increase, and that the patients and the clinicians will use it more, because it's not that we don't have a treatment is a good answer to not do anything, because if we don't have enough patients, then we will never do a clinical trial. So it's really that's the circle.
And that's going to me a related question, Armel, that we discussed. As a rare disease, you're facing extra complexity because the disease is inherently heterogeneous and very complex. First time I discussed with Nathan, and mentioned VCP, I was coming from the ALS field, right? And I think a lot of people still know VCP from ALS. And so how do you handle or how do you deal with that extra complexity of bringing awareness on the disease, as a foundation? I know it's hard, but maybe you can point us to some activities that the foundation is doing to bring awareness as a foundation, but also what's the reaction to drug developers? I'm sure you interact with industry. How do you approach the conversation to try to attract them to the disease? It's a target for VCP, but it's also a protein involved in other disease. So there are means, I'm sure, to create some interest from them.
Yes, we have. I started 18 months ago at Cure VCP disease, so I'm just at the beginning. But the interaction I have with the industry, they show an interest. First, because we are ultra rare, so if we want to test drug, we have a natural history study. We would be able to have endpoints and a trial in only 2 years, which is a big advantage. So I think it's a good opportunity for them to do a clinical trial in a small population, well defined, with the natural history study, and get some points, get the orphan drug approval or label, and then expand to other degeneration that are larger or maybe more difficult to assess. So the fact that unfortunately our patients decline fast is seen as a good thing for clinical development. Not at all for our patients, but that's it's for now well received. We have also compounds that are developed mainly for oncology. So those are inhibitors, so they might be applicable for repurposing in neurodegeneration, but since we don't really know which type of compounds we want, so that's the reason why we developed these organoids, so that we can screen on phenotype more than on the protein activity. Which might not be the knowledge on the function and the relation between the ATPase and the function might not be in fact the key of everything, but more the protein interaction with the cofactors and or really activate certain parts of the protein or inhibits only one domain and not the other. That's it's for chemists, it's very interesting. VCP is very interesting for them.
Great. Taina, I don't know if you have other questions. We have a few things on the Q&A from the audience, so I'll go there unless you have a burning question. Okay, I'll get there. So I mean, it's goes into that conversation about the heterogeneity of the disease. What is the most common misdiagnosis across the population with VCP variants? Or maybe if I want to change the question, talk to us about the misdiagnosis, patients that have VCP disease that goes undiagnosed, and patients that are, and vice versa, patients that are misdiagnosed with the VCP disease and it's actually something else like, ALS or another neurodegenerative disorders.
So we have a lot of misdiagnosed in LGMD or myositis in the muscular population. I mean the degeneration of muscular population. But we can imagine that in all the dementia we have a lot of misdiagnosed people. Even in Parkinson, we might have a certain number of misdiagnosed because they don't do clinical genetic testing, and in Charcot-Marie-Tooth, there's a lot of patients that are classified as Charcot-Marie-Tooth, the 2 type, and they don't go further. But the CMT2Y is really a classification of VCP patient with Charcot-Marie-Tooth. And we might have patients there that don't know that they have the VCP mutation and were just diagnosed as Charcot-Marie-Tooth. And the VCP is highly expressed in the cerebellum and in the Purkinje cells. So all these disease linked also to Purkinje degeneration might be linked to VCP and might be misdiagnosed also.
Do you have a comment, maybe, Terina, on misdiagnosis, or I'll jump to the next question?
Well, just actually a follow-up comment, because again, I think that you've done such a wonderful job giving us a representation of the complexity, and also I want to commend you because it is so complex, but you made it very understandable. And I think that as you mentioned the convergent cause, divergent effects, complexity of misdiagnosis. I think that the approach that you discussed, the integrated approach, is a really thoughtful way to address those really complex questions. And I also think that your thoughtful and considered approach to do the screening and hope try to get learnings from other areas like the oncology space is really key. And then of course the work that you've done in the last 5 years has been really impressive, and then preparing the patient population for eventual therapeutic testing is really commendable. So I'll just leave it there so Alex can bring in another question.
Yeah, I will actually have
just if I can add a comment on that. Our disease is really a neuron. So it can be any type of part of the brain or peripheral neuropathy, and we have muscles and we have bones. So if you think about rare disease that have all these three, neuro, muscle, and bones, we are not the only one. And you can be sure that in those kind of disease where they have heterogeneity, where they have patients that have bones manifestation and other do not, that have bone manifestation and neuro manifestation, there might be a VCP mutation modifying their own mutation and triggering in one direction rather than another. So that's also where the really screening larger or trying to understand where VCP could be a gene modifier could add a lot to the VCP population, and help also other populations that have these three components: neuro, muscles, and bones.
Thanks.
Sorry, I interrupted you, Alex.
No worries. I have 2 additional questions that are related. One of our attendees is asking about the clinical manifestations, and given the heterogeneity again, are there patients that actually don't get this disease symptom? Or maybe can you comment more broadly on the extent of the clinical manifestation severity, from less severe forms or asymptomatic to extremely severe and fast progressive form? And also that brings me to a second question about the clinical outcome assessments and how do you measure patients, and I'm sure it's extremely difficult with no dedicated, clinical outcome assessment specifically catered to VCP to handle that, I'm guessing, heterogeneity in terms of clinical symptoms and how to best measure patients. And I know the foundation is doing a lot of work, and so yeah. So first, are there any asymptomatic patients? And then 2, how do you handle measuring patients properly?
So I think it's there are different level of difficulty for that. First, the clinicians are not really neuromuscular disorder clinicians are not really aware of VCP disease. So they have patients coming to them and they don't have the exact diagnosis criteria for VCP, or they don't even know the standard of care, most of them. So they have a patient coming and they will try to identify muscle weakness, for example. So they will characterize them as IBM patients. And then a few years later, they measure the activity of motor neurons, and then they say, oh no, you are not myopathy, you are more Charcot-Marie-Tooth, or then they move to, no, you are not, you are more ALS. I think one of the, and you see patients going from one diagnosis to another because of the evolution of the disease, and because there is few knowledge on the real characterization and the evolution of the disease. So that adds also to the complexity, and the fact that clinicians try to put our patient in boxes, in the existing boxes like ALS, Charcot-Marie-Tooth, or IBM, but maybe VCP patients are another category with an evolution of the manifestation, and they do not stick to one diagnosis. So that's a very difficult point for the clinicians. So I think the natural history study has to go on, has to increase, even though we would be able to have endpoints already after a year or two, let's say 2 years. We should still continue to expand to all the different type of mutations and expand the natural history study.
Great, thank you. I have an additional question on the clinical aspect. I mean, you mentioned pediatric patient, and we actually have someone who has a pediatric patient diagnosed with a VCP mutation, and she was asking, is it okay? I mean, can she subscribe to the registry? Is that a population that you are studying right now? And if so, are you collecting fibroblasts and everything else?
So yes, we are an organization dedicated to all VCP mutations. So pediatric, late onset, dementia, neurodevelopmental issues. So the pediatric population shows there is a part of them that show muscle weakness, but not all of them. And it's more characterized by autism, learning disability, speech delay, and in some cases seizures. And we really need to it's new because up to now it was not known that VCP could be involved in neurodevelopmental disorders. But what's interesting is that you can see really the link scientifically between the involvement in developments and then degeneration for neuro. But yes, we are. They are very welcome to contact us. We try to do all type of mutations, from developmental to degeneration to dementia, everything. And we are referring those patients that come to us with pediatric patients. We refer them to the right pediatrician, and it's important to follow them for the muscles, because since the muscle manifestations are usually late onsets, they are in pediatric also, more in adolescent, but they are basically defined as late onset. So we need to follow those patients along their life. And if they agree, I know for toddlers, we have patients with toddlers with VCP mutation. It's not easy to give blood, but having samples to be able to identify maybe biomarkers before symptomatic and during symptoms would be really helpful. So that's also something we are looking for. Pre-symptomatic people who know they have the mutation but don't have yet muscle degeneration or dementia.
Thank you. Taina, do you have a final question? Maybe one question and one comment to end.
Right, thanks, Alex, because I do want to use this opportunity to bring something together, because it really does emphasize a lot of what Armel was just saying. So I think that again, the way you've outlined the complexity and the approach is really key, and it's actually ties in well to some areas that we're scaling into in 2024 and beyond at CPAP through the rare disease programs. Because we're trying to take an approach that looks at sometimes families of diseases, so that we can try to have a greater group and more diverse stakeholder engagement to pursue COAs or improved patient reported outcomes, et cetera. But then we're also trying to take an approach where where we can have a better understanding at the progression and mechanistic level when we're looking at something that's more specialized. So, for example, we've got some efforts coming up looking at pediatric seizures, and that's where we can see if there's ways to harmonize and synergize some of these efforts. And of course, everything is data driven. So I think it's so important that you have your natural history study, your registry, and it's just really great to see what you've done in 5 years, and really will be very exciting to see where you go next.
Serena, thanks, Serena. That was actually like a good segue to my last question. I mean, we've been working with Armel and the Cure VCP Foundation to get data in RDCA-DAP. We're gonna have some registry data coming up soon. It's under negotiation right now, but we're very hopeful it's gonna arrive. Additional data coming directly from Cure VCP as well. So we're gonna start building and aggregating data. I know it's early, but maybe what's your hope for the first thing we could try to tackle with that building data aggregation in RDCA-DAP? What unmet need you think we're gonna be able to start working on in the near future?
Well, I think the biomarkers, trying to find biomarkers that would be across rare diseases will be very useful. And the next part would be, my dream would be to have a kind of modelization of the disease progression, but that would imply to go across diseases and have the disease progression for the dementia. That could be maybe across different type of mutation for the dementia, and have disease modelization for more the muscle part. We have also patients that are more HSP, that are more type ataxia. We have some Parkinsonism, so that's all across diseases, and modelization. Maybe we can benefit from larger population and try to see if it fits with our smaller population, if there is a discrepancy between other type of Parkinson disease and VCP Parkinson disease, or and try to tailor a little bit that. So that's my hope.
Great, thanks so much. And perfectly in line with what we do at CPAF. So that's why we brought you to this webinar. Thanks again for your presentation. It is the last webinar of RDCA-DAP of the year, but we'll have new webinars upcoming next year. I just want to end by thanking you again, Armel. I also want to give a big kudos to Nathan and Alison Peck from the foundation. The first time I met with Nathan, I think, was 2 years ago. He's a force of nature, but it's really, really impressive, everything that was achieved by the foundation in the span of 2 years. You showed it, but I want to re-emphasize all the achievements that was only even more boosted by your arrival, I think 1 year ago. So thanks again for the presentation. Thanks everyone for attending the webinar. We'll see you next year.
Yeah, and I want to thank the patients. They are engaged, and it's thanks to them that we are there.
Absolutely.